Hello, I am extremely grateful to the ESPID Scientific Committee for recognising my work and research outputs, which I've been involved with during my PhD and my subsequent postdoctoral period. I'm currently a clinical postdoctoral scientist in the Department of Zoology at the University of Oxford, and I'm a locum consultant in paediatric infectious diseases at St George's Hospital in London. I would like to give you a brief insight into what I do and how it has been impacting infectious disease prevention. I chose to study genomics because I knew it would impact my clinical practice in future. In the last 5 years, I've advanced my genomic epidemiology research career rapidly, from entering the field of genomics to making significant contributions to public health. The meningococcal vaccine Bexsero was introduced into the UK infant immunisation schedule in September 2015, at the same time as I started my PhD, addressing questions of how well this vaccine will work on a population level. I learned microbial genomics and bioinformatics, developed 2 novel genomic analysis tools, and have identified 3 clinical applications to date. The first is a vaccine traffic light system, now called the MenDeVAR Index, which was applied to individual cases and developed in response to a university outbreak, where public health colleagues sought supporting information for deciding which of 2 licensed vaccines to give to close contacts. I recognised that we needed better methods of translating genomic data to inform clinical decision-making. Hence, I developed the MenDeVAR Index, now routinely used in the NHS in Scotland. Secondly, these tools have been used by academics and reference laboratories globally for population surveillance, which contributes to vaccine policy decisions. I performed UK national surveillance of over 3,500 UK meningococcal isolates using my genomic analysis tools, and now collaborate with reference laboratories globally to do the same across 16 countries and 4 continents. Thirdly, my tools are transferable to different bacteria. For example, the gonococcus, a close relative of the meningococcus, and a global health threat due to increasing AMR gonorrhea. In the search for a gonococcal vaccine, my genomic analysis tools are now being used for antigen discovery for gonococcal vaccines. All this work was possible due to the large collection of annotated genomes and user-friendly infrastructure of the PubMLST database available here at pubmlst.org. All the analysis tools I developed are publicly available on this site, which has over 20,000 new users a month. This platform also provides a means for the global public health microbiology community to find important information about genomes, and the way we communicate this is continuously improving. The meningococcal deduced vaccine antigen reactivity index, or the MenDeVAR index, was developed to combine complex genotypic and phenotypic information into a simple, evidence-based output for frontline healthcare professionals. You can see here an individual genome record that you will find on PubMLST with that unique identifier, some information about where it's from and its serogroups. And here you see the vaccine selection, where we show the reactivity for both Bexsero and TrueMember. And this is showing how well the vaccine antigens in the isolates match to those in the vaccine. And where these antigens have been tested experimentally, this is compared and demonstrated here, showing all the references for you to assess cross-reactivity for frontline users to quickly assimilate information with the evidence base apparent to them. As many reference laboratories are now sequencing meningococci in real time, this information can be available hours after sequence data is generated. Users can analyse their genomes by uploading them, like this, as a record onto the database, or using a straightforward plugin with a copy and paste of their genome sequence data. In the last 18 months, I've also been involved in the process and analysis of the largest longitudinal carriage collection of meningococcal isolates, taken from over 15 years in the UK, with over 67,000 participants, all 15 to 19-year-olds. As meningococcal disease has steadily fallen since the late 90s to now, particularly serogroup B, we studied whether there was a change in carriage over this time, as carriage is the prerequisite to invasive meningococcal disease. We found that carriage rates by age had fallen significantly between 1999, shown in blue, ranging from 10 to 22%, compared to 2014-15, in purple here, ranging from 4 to 9%. Interestingly, however, the odds ratio for carriage for each age group remained unchanged, increasing from 15 to maximum at 18 to 19 years. The participants were also asked to complete a risk factor questionnaire regarding smoking, kissing, and pub club attendance. The fall in adolescent carriage prevalence between 1999 and 2014-15, shown in the left and right respectively, was concomitant with measurable changes in social interactions between adolescents. Participation in carriage promoting behaviours fell by 47% for smoking, 17% for intimate kissing, and 37% for pub and club attendance between those two periods. Our observations have global implications for modification of public health interventions and vaccine policy to reduce the morbidity and mortality resulting from an invasive meningococcal disease and other pathogens residing in the respiratory tract. Thank you very much.